Good afternoon, guys. Last class we talked about transcription, which was we made a copy of one strand of the DNA and we turned it into messenger RNA. Then this next step, the one we're going to talk about today, translation, we're going to take that messenger RNA and we are going to turn it into a protein. Here's our learning targets today. Please pause and write these down on your notes sheet that has the Ranch View High School header at the top. Our we will, so we will describe and model the steps of translation, the process of turning mRNA into proteins, and explain how it is important for gene expression. In the I will, you will explain to your partner the purpose of translation in gene expression. Our essential question is, how are genes expressed? Now, when we talk about translation, we're talking about taking mRNA, turning it into a protein. Well, where does this happen? Well, mRNA leaves the nucleus because it's the only thing small enough to fit out of the nucleus, right? The whole reason we have mRNA is because DNA can't get out of the nuclear membrane. So that's the mRNA's job. It's small enough. So it leaves the nucleus and it connects to a ribosome. There at the ribosome, the mRNA code is read and translated into a protein through a process called translation. Now let's talk about the role of the ribosome. We don't talk a whole lot about it, but it's incredibly important. We're not going to get into all like the super big details about the ribosomal sites. There's an E site, a P site, an A site. It's not important to us. If you find some videos online to learn more about translation, they might go into more detail. Just know that you don't need to memorize it to that level of detail. What you do need to know, though, is that the ribosome is kind of like the bread of a sandwich, and it's going to clamp down on either side of the mRNA when it arrives to the ribosome, kind of just grabbing onto it. And if you've ever fed paper tickets into a machine at like Chuck E. Cheese, and it just like grabs the paper and pulls it through and counts everything, that's kind of how the mRNA is going to work with the mRNA. It provides a site for protein synthesis, and when the mRNA leaves the nucleus, the two ribosomal subunits, they'll come together and they'll hold onto the mRNA in place for translation. I did put this slide a little earlier on, so if you'll like just go down on your notes page a little bit, the role of the ribosome, and then come back up to where you were before. I don't want you to miss your notes. Now let's talk about translation. We have our ribosome. It's grabbed onto the mRNA. It's pulling the mRNA through the middle of it and reading all of the mRNA bases, the nitrogenous bases of the mRNA. And the way it does that is by organizing that information into 3-letter blocks called codons. Codons are very important, and they work kind of like reading a word. So the ribosome takes groupings of 3 letters and translates those into proteins. And here's how it does that. The transfer RNA molecules, the tRNA, it acts as an interpreter to that mRNA codon sequence. So it is going to say, oh, okay, I see that I have an A, a U, and a G, and that's my 3 letters. Well, I have the matching letters, or not matching, but the complementary letters. So if the mRNA strand says AUG, well, what goes with AUG? Well, that A becomes U, the U becomes an A, and the G becomes a C. So when I find the anti-codon that says UAC, you see right here where my mouse is, UAC, it is going to attach itself, just like you see here in the image, to the codon on the mRNA. Now, this little grouping of 3 right here, not on the mRNA, is called an anticodon. And it's held by this little clippy looking thing. That's the tRNA. And on the other end of the tRNA, and you can't really see it in this picture, but you can kind of see it on this middle tRNA, is an amino acid. And what happens is, as a new tRNA connects, It's going to add its amino acid to this string, and over time, what you get is a long protein. Well, let's talk about codons real quick, because they're complicated. And last time I counted, there were 26 possible amino acids and a whole lot of possible combinations of letters. And there's two ways that these codon charts are represented. All of this is on the back of your paper. And anytime I'm asking you about codons, I will provide you a copy of the codon sheet. You don't need to memorize this. I had to memorize it in college. Don't recommend it. Not worth it. How do you read it? Okay, we start with the first letter, just like a reading book. We're going to go left to right. So we start with our first letter, and on the left side of this block, we will find the block that matches that letter. So if our first letter is an A, then we'll go over here to A, and we go to our second letter. Well, our second letter is a U. Okay, so we have to stay inside of this block for A here, which is this whole area. Do I have like a, can you make a laser pointer? There we go. Okay, so I have to stay inside of this A block. I can't go outside of it because my first letter is an A. And what I have to do is go up here to my second base and find the one that matches my second letter. In this case, that is a U. So we follow our U down to where our A block is, and we know, okay, it's somewhere in these four. Then we are going to find our third base and match it. So in this case, it's an A, a U, and a G. We found our A, we found our U, and right here is AUG. This is methionine. It's also our start codon. You will get really familiar with seeing AUG because it should be at the start of every codon sequence that you're supposed to translate. Now, what we have in the inside here is the amino acid. Like I said, methionine. And you can do this for any combination of three-lettered codons. So if you have a UUU, well, you'll find your U, you'll find your U, you'll find where it's a U again, phenylalanine. Cool. If you have a GAG, you'll find your G, you'll find your A, you'll find your G right here, glutamate, right? So you just process of doing this over and over again, it can be a little tedious, but it's not hard. Now, there is another chart as well. And this one, I think this one's a lot easier. I prefer to use this one. Different teachers, different scientists have different preferences. This is my preference. And this one's really simple because we just start in the middle and follow our way out. So if we have here an amino acid is CUG, we put our finger right here in the middle and we go to the letter C. And then you see how these lines here are a little bolder? That kind of works like a fence. So I need to find the nearest U that's touching my C block because I need to go C, U, G. So I would go from C to U right here. Then I would find the base of G. Oh, look at that. And then follow it out. It's leucine. And this again works with any combination of letter. If I have AUG, like we said before, we go to A, we go to the U that's touching it, we go to the G that's touching that. Boom, methionine. Really straightforward. One last thing for you here. This is a codon activity. I want you to take this strand. It says break this strand into three-letter blocks called codons. It's already broken down into three-letter blocks for you. What I would like for you to do is use your codon chart to identify what amino acids go here. You don't have to write the whole amino acid. You just need to write the first three letters of it for now. That'll be enough. So for example, here we start with UAC. If we go back to our chart, we go to UAC. That's tyrosine. So you'll write TYR. UAC. TYR. So you'll put a little TYR here. And I want you to work through the remainder of these. It's just a little practice and put those in your notes. I'll leave this up here on the board for you. A little bit easier to read this one. Now, what do you need to do next? That is the big question. Once you have completed your notes, I would like for you to write your short constructed response, your SCR, and you just read this, answer it inside the box, turn it into the tray. This will be a grade. Please complete this by the end of class. Then, once you have done that, I want you to complete the gene expression and bird activity. All of these papers should be or should have been on the front desk that you normally pick up your papers on. And read the instructions. Here it says the species of birds has only one chromosome with 6 genes on it, which are used to determine their 6 traits. Your job is to analyze the genes of its DNA and determine what traits the organism has. And here we have a DNA. And you'll translate it into mRNA, which means we can't use T in this mRNA sequence, right? So, for example, here, this TAC, it would become AUG, and so on. Then you will use the codon chart. Probably easiest if you just go back in the video where it's on the screen and use that codon chart to find your amino acids. Then you'll use this amino acid sheet sequence here, follow these, and it will identify your traits. Follow the instructions on part 2. Draw your bird. Once your bird is drawn and colored, the colors are up underneath my pencil sharpener at the front of the room. Once you've done all of that, make sure to turn both of those papers in. That is all that you need to do today. Apart from that stuff, I do want to talk really quickly. I put some messages up on Parent Square, but I wanted to just remind you guys about tutoring this week. I'm not here today. I'm not going to be here on Friday. I will have tutoring for you before school on Wednesday if you need it, and after school on Thursday if you need it. Just let me know what you need. Send me a little email so I know. That is when our tutorings are this week. There are no other tutorings. All right. Have a good time. See ya.